Our next speaker is Dr. Becky Crean, Executive Director of Clinical Development at Ionis Pharmaceuticals, who leads the clinical development of Ionis's ASO programs, including HALO study that she's going to talk about today. Becky?
Good morning, everyone. First of all, thanks to FAST for inviting us to be here. I think I can speak on behalf of all of us at Ionis and our partners at Biogen, and we really are thrilled to be here today to talk with you about our program. And you know, we've been coming here for a number of years, and it really is remarkable each year we come to see such tremendous advances made, not only in the science, the research science, the early science, but also in the development for treatment. So it certainly does energize all of us back home at Ionis and makes what we do really important work. So thank you for having us. I'm going to spend the next about 15 minutes talking with you about our program and some updates. So there's going to be three things I'll cover today, and one is really just our approach to this developing a treatment for Angelman syndrome. And so I'm going to spend a little bit of time talking about our compound, how we decided on it, what our approach is to our trial design, and then I'm also going to talk about some really high-level findings and observations we've had from the ongoing trial itself, a little bit about safety and tolerability of what we're seeing with the compound. And then finally, we'll talk a little bit about, from this early read in the Part 1 trial, we'll talk about the impact that we're seeing on some of the clinical measures. So I'll just preface this by saying this is very high level and very preliminary findings, but we are encouraged by them and excited to share them with you today. So before I get into all of that, I want to take just a quick step back and talk about really what are the goals of this program. And so you've heard us talk before about the different stages of clinical development. And with each stage and with each study, they're designed to answer a really specific question. And our job is to answer that question as efficiently and quickly as we can, and then move on to the next stage and answer the next set of questions. So for this trial, this is an early phase 1/2 trial, and it does have a very specific question, and it really is to answer the question of whether this compound, ION582, is safe and tolerable in patients with Angelman syndrome. That's first and foremost is safety. That's the first thing you answer when you step into the clinic with a compound. So that's really what this trial is designed to tell us and answer that question. And we're doing that with two different parts. Part 1 is what we're calling the MAD portion. That's the multiple ascending dose portion. We're looking at several different dose levels, several different dosing intervals in different age groups. That's really going to give us information about how safe and tolerated the drug is over a really short period of time. It's 3 months of dosing and in a very small number of patients. Part 2 is really designed to tell us information about the compound, safety, first and foremost, over a longer period of dosing. It's 12 months, but it also starts to give us a glimpse into some of the clinical functioning and if the compound is having an effect clinically. So what is this ION582 and why did we choose it? So I'll spend a little bit of time talking about that. And so you've heard a lot about ASOs, and I'll talk specifically about this one. So the one we've chosen to go into the clinic, it's really designed to stop the silencing mechanism on the paternal UBE3A brain. You know about this approach. All the ASOs in the clinic are using this approach. We're trying to stop the antisense mechanism on the paternal allele. And what that does is that'll release the ability of that gene to produce UBE3A protein in the brain, which we think is important to correct. And so this particular compound, ION582, is what we call a 2'-Mo antisense oligonucleotide. It's a mouthful. It's a certain type of modification to single strands of nucleotides. So with this approach, what we do is we change the two ends of this single strand of nucleotides and we leave regular DNA in the middle. And what this chemical modification does is it allows for cleavage or cutting off of that antisense transcript that's stopping the paternal allele from from releasing UBE3A. So that's the approach we're taking with this particular ASO. But not all ASOs are the same, and so there's different chemistries and different chemical modifications that you can make to get different effects. And so different chemistries have different properties, different potencies, different duration of actions, different safety profiles, they're tolerated differently. And so what we particularly use 2' Mo ASOs when we're going into CNS disorders because they have greater potency and for reducing the target RNA, and they tend to have greater tolerability, meaning how well patients can tolerate the compound. And so it's for those reasons that we choose Mo chemistries to go into disorders that are affecting the brain. We have a lot of experience with this at Ionis. We've been doing this over 30 years, and for the first 30 years of our existence, 100% of our focus was on developing these ASOs and learning more about them. So we have a rich history and knowledge base back home to do this, and we've had some great successes. You can see here things that are in the clinic in diseases like SMA, ALS, Huntington's disease. Thousands and thousands of patients have been treated with this particular kind of chemistry for this ASO. And so some of them have been approved. All of these are in clinical trials at some stage or another. So we have a good record with this, and we think we know this type of chemistry very well. We feel comfortable going into the clinic with it. But how do we choose Ion 582 in particular? How did that come to be in the life of ASOs? And so this one, again, was really chosen for its safety and tolerability that we saw early on in animal studies that we thought would translate well into humans and into the clinic. So we start with a really wide net, shown here as a funnel. We screen over 3,000 ASOs early on in the cells and cultures, and we look specifically for ones that do a really good job of binding to that antisense transcript RNA, the one that we're trying to knock down. So from there, we start to whittle down the ASOs that we're looking at based on different characteristics. So from there, we pick the ones that are best at binding to that target RNA, and then we start to look at which ones are the most effective in different models of Angelman syndrome. So we look at transgenic mouse models. Those are where the human gene is actually inserted into the mice. We look at Angelman syndrome mouse models. We also look at their effectiveness in iPSC neurons. So a number of different steps that we go through to see which ones seem to be the most effective. And then from there falls down into the funnel fewer ASOs. And so we continue our exploration of this in our research studies. And ultimately, we select a handful that look safest and most effective to take into larger animals. And in these studies, we usually use non-human primates. They're the closest biologically to us and give us the most rich information on what it might translate like in the humans. So Ion 582 was chosen out of a number of thousands of ASOs that we looked at and really identified as the best one to take into the clinic. And we've seen this through multiple animal studies, and we've also tested it at much higher doses and greater frequency than what we're seeing in the HALOS trial. So we feel like we have a really good safety profile and really good safety margins to go into the clinic. So that's a little bit about how we came to be with Ion 582. And based on the properties of this compound and the mechanism of action for and the genetic cause of Angelman syndrome, we think that this compound really does have the potential to treat and be effective with all individuals with Angelman syndrome. We think everybody could benefit from this compound. So you have to test that. So we're starting first in the HALOS trial, the one I'm going to talk with you about today. And it is a pretty broad population that we're looking at. We're looking at toddlers down to 2 years old, all the way up through adulthood, 50 years of age in this first clinical trial. We're looking at 2 of the 4 genotypes, deletion and mutation. And from there, what we're going to do is really use this data that we get from the HALOS trial to help inform on using this drug in additional, in the rest of the population. So it has to be data-driven. So that's why we're doing this early trial. We're going to get a lot of rich much data from it that will help us plan and think about next steps for broader populations. I mean, we've talked about this at this meeting for many years about the theoretical risk of overexpression with a UPD or ID genotype. It hasn't been tested before, but the data we get from this trial will help us understand what that might look like going into the clinic. In those really young populations, infants, important to look at, and all the different genotypes as well. So it'll be data-driven, but we do think that we'll get there and that this is a compound and an approach that will work for everyone. So I mentioned that this trial is a small trial. It's our first in humans, but it's got a broad reach. So we're spanned over 6 countries. We're in 11 different sites across these 6 countries, and everybody's been able to contribute to enrollment up until this point. So the design of the trial, this is a really important trial for us. As I mentioned, it's the first one in humans, people, and so it's designed to answer that very specific question that I talked about, which is really primarily safety and how well the drug is tolerated in individuals with Angelman syndrome. So that's our primary goal with this trial. And so we have 2 parts to this. It is a phase 1/2. The first part, what we call the MAD, as I mentioned before, is we look at 3 doses, different levels, different intervals in the first 3 months. From there, patients roll into what we call part 2, which is the long-term extension. That's dosing with Ion 582 for an additional 12 months, looking at different intervals as well. And everyone in this trial receives drug. There is no placebo in this trial. As I mentioned, we're looking at a broad population in this early trial, males and females, 2 to 50, deletion and mutation. And during the trial, we're giving a lot of tests to gather as much information as we can in this early phase. So we're looking at things that are important to families, things that Angelman syndrome affects, like walking, talking, sleeping, seizures. We're measuring all of that in this trial. Some are done in the clinic, some are done by the parents, some are done at home. So we're trying to gather as much information as we can so we can inform on the next decision. So this is a schematic of what it looks like. As I mentioned, part 1 MAD, 3 months, 3 doses, and we group them differently depending on the dose and the age group. So how we've depicted them here is just the different age groups and buckets that we're looking at. 4 to 17-year-old looks at this first 3 months with different dose levels. We're also looking at these dose levels in 2 to 5-year-olds, and then also in adults too, because it may be that different age groups respond differently, so we want to make sure that we look at them all separately and then look at them in their totality as well. And so the goal of this one really is safety. Then everybody rolls into, once they finish that first part, they roll into the part 2 long-term extension. And again, here really it's treatment for a longer period of time, 12 months, different dose levels, different dose intervals. And what this will tell us is really start to help us find what the right dose is and dose interval that might show the most clinical benefit so that we can then take that into the next phase of development. So this is a little bit about the design, our approach. I told you we were going to talk about 3 things today. We just covered thing 1. So now what I want to do is move in and tell you how we're doing in the trial. We've come for the last couple of years and given you updates as we can. And so now I want to share with you that this trial, I'm happy to report, has completed enrollment. We've been able to do that over the last year. All 11 sites have been able to participate and enroll participants and family into this trial. And so I can't say that without acknowledging the role that you all have played in getting us to this milestone. Everyone sitting in the room, whether you participated or not, you did in some way, whether you know it or not. All of the families that have participated in the trial, and of course the patient organizations that provide you with the information to help you make informed decisions on whether to do this or not. So thank you for that, and we've accomplished a lot this year. So because we've completed enrollment in MAD, we'd like to share with you some of the early preliminary findings that we've been observing over the last year and a half in this trial. And this is from the part 1 MAD part, that part on the left, the 3 months of dosing. I just want to preface this by saying these are early data from an early data cut. We cut the data at the end of last month, so it's a partial data set. And it doesn't represent the final data set. So this is starting to give us a signal into what's happening in the trial. But the goal is to complete the longer-term dosing and look at all of the data so that we can get a good idea of what's going on. This just gives us a snapshot. And so what I'll show you in the next couple of slides is some of those early findings, but everything, all the data is pooled. It's not being broken out by the age groups or the dose levels or the intervals. This really is a very high-level preliminary look at this. So I can't emphasize that enough. So I told you that we're looking at different dose levels, different age groups. So far, when you look at it at a very high level, this compound, ION582, has been well tolerated at all dose levels that we've tested in this trial. So as part of the design of this study, we have what's called an independent safety monitoring committee that reviews data on an ongoing basis. Early on, they look at it very, very frequently as we're getting into new doses, new age groups. They review the data on an ongoing basis, and then we have meetings with them, and they report their thinking about it, their recommendations, their findings. So we've been doing this with them for quite a while, and so to date, no concerning safety trends have been observed, not only by our independent data monitoring committee, but also our own internal monitoring committee and the CRO's monitor that we're working with as well. So there have been adverse events. It's not unexpected, but so far, the ones that have been reported during the trial, the majority of them have really actually been consistent with the patient's medical histories, symptoms associated with Angelman syndrome. We've had some also that are related to the lumbar punctures, but by and large, fairly mild, fairly few, and we're very encouraged by the initial read on the safety of this compound. We also take we look at a lot of measures in laboratory assessments, in CSF, in blood, in urine, and so far to date, all of those have been clean and no safety signals in any of those laboratory values. In the panel on the right, you'll see kind of who these families and participants are in the trial, and we've grouped them by age. So you'll see that in the 4 to 17-year-olds, we've had 28. We've had about 9 adults in the trial, and then about 14 of the really young kids as well. So in total, we've enrolled 51 participants and families in the trial. You can see the age range there is anywhere from 2 up to 34 years of age. Had a fairly even split of males to females, although a few more males than females. And then primarily deletion, about 85% and about 15% mutation, but I think that's expected given the prevalence of the genotypes. So it's a little bit about who's in the trial and from a safety perspective, what some of these early signs are we're seeing. And so now I want to talk a little bit about some of the clinical and biomarker observations that we've had. So I won't go into detail on this. We're using EEG. It's an important biomarker, we think, for all the reasons you've been hearing over the last 2 days. Kemi just talked about it in the last talk. And so we think this is a good tool, a non-invasive, relatively painless, maybe more for the parents, to measure brain activity. We can measure electrical activity in the brain from millions of cells that are active in the brain, and we can get a good idea of what's happening in the brains of individuals with Angelman syndrome. And we know, you guys see this is a great, I'm so glad this research was done. You see it all the time, and it's important. That's why we show it a lot. This is just the difference of EEG activity on delta activity that you see in neurotypicals, which is on the left in the black lines, pretty flat, little squiggly, and then those of Angelman syndrome on the right, where you see those really pronounced delta waves. And so because of additional important work that's been done and published recently, we now are starting to understand what EEG might mean in terms of a clinical sense. And so there was some publication that came out a couple of years ago that started to link the dot between EEG activity and clinical functioning. And so this is just a correlation graph of EEG delta waves and how it correlates to cognition on the Bayley. We know about the Bayley. So this is just saying all those dots are individual patients, all all hugged on that diagonal line, which is just a good way of saying that EEG seems to be strongly correlated with clinical function, particularly cognition, as we measure it on the Bayley. So that's encouraging. It's not going to replace a clinical test, but it certainly can tell us if things are moving in the right direction in the brain as well. So in our trial in HALOS, we've had the ability to look at some really early signs on our EEG. We look at EEG at different time points in the trial. And so from the data that we've looked at so far up through October, we can see that a majority of the patients that we've done EEG on, about 70% of the subjects have shown a reduction in this slow wave delta region that we're talking about. And so we look at that compared to baseline, and this is taken 1 month after the last dose in that MAD portion. I told you there were 3 doses. So this EEG is taken about a month after that last dose. So we're not talking about magnitudes here. We're just showing that percentage of subjects who are starting to show a decrease on the delta activity. This is seen, we see this in animal studies, so it's nice that we're seeing the translation from what we saw early on in animal studies now into the clinic. We think that's encouraging. And in addition to delta, we also look at theta, which is the faster frequency rhythms. And so we also see the majority of our patients so far have shown an increase in this theta activity. And again, this all suggests that the EEG is improving, and we know there's a correlation with EEG and functioning on clinical measures. So we find this encouraging. It's early, and we don't have a lot of details about it. There's a lot of rich data to be dug into once the trial's complete. This is an early read, but we think this is encouraging. So we also look at clinical measures. We take a number of we do a number of clinical measures in the trial. 2 of the ones we look at are the CGI, which you've heard about, different versions of it, and also the Bayley. So I won't go into too much detail, but the CGI this one was developed by Roche, called the Symptoms of Angelman Syndrome Clinical Global Impression of Change. It looks at clinical functioning and change in 9 key domains of functioning with Angelman syndrome. They're anchored. And then also the Bayley. I won't go into the Bayley. It is a direct assessment of functioning. It's sort of the gold standard in the field on how we can look at a direct measure of functioning. And so we're doing that in this trial as well. And so on both of these measures, again, early signals are showing some clinical changes observed a month after that last dose in the MAD portion of the trial. On the top bar, that's the CGI, so we see a majority of patients demonstrated some level of improvement. You know, it's rated on that 7-point scale. Lower numbers mean better. Some level of improvement on the as rated by the clinician on the overall CGI score. So, you know, you can break it down into those 8 domains, and then the 9th one is just overall, how are they doing? So that's what we're showing here, is that clinicians say overall they've noticed an improvement as they're rating these kids, and that it was considered meaningful. Now that's a relative term. We talk a lot about meaningfulness, clinical meaningfulness, meaningfulness to families. So this is just the clinician's interpretation that they thought this was a meaningful change. When you look on the bottom, that's the Bayley. We looked at the total score in the Bayley. There's a lot of ways to slice this pie. There's different domains. This is just looking at the overall total raw score. And what we see there is that the majority of the patients so far have shown some level of improvement on that total Bayley score. Again, we think this is encouraging. It seems to be greater than what you might expect in the natural history that we've seen, and you saw from Kemi's talk that it remains relatively stable over the lifespan, so we're starting to see some improvements on that as well. So we think this is encouraging. This is early days. We need to follow this up. We need to complete the trial. So what happens next? We have this encouraging data. We've completed enrollment. So where are we going from here? We think we have another busy year coming up. And so the primary goal for us is really to focus on completing the data accrual in the HALOS trial and analyzing that data from part 1. So we have these preliminary findings that ION 582 has been safe and well tolerated so far. We're starting to see early encouraging clinical signs of change and improvement, but we really need to follow that up with a full data set. We need to dig really deep into that data set to look at all the nuances. And with the final data cut and readout from this part 1, we expect to occur around mid-time next year, so mid-2024. So participants continue on in the trial. As I mentioned, we have a part 2. It's much longer term dosing. So participants will continue through that part 2 phase all of next year. And that'll really provide us another rich data set, still looking at safety, but also really trying to confirm some of those early benefits that we're seeing in the first part of the study. So that'll be an important thing that continues to be collected through next year. And then finally, we still continue to collaborate on a pre-competitive basis with our other industry sponsors. We've got some natural history study data that we still need to analyze that's being written up. Some of it's already been published. You've seen some publications come out of Friesa. There's more data to be collected there, so we'll be working with our partners on that as well to keep getting that data out there for the community that we can learn from. So we've made a lot of great progress, a lot more work to do, but I just want to stop and say thank you to the HALOS participants in particular. I don't know if any of you are here, their families, their caregivers. These aren't easy decisions to make to be in a clinical trial. They're not easy to do. We ask a lot from you, and we truly do appreciate your time and effort and your commitment to doing this. And also to the 11 study sites, it's a big load for them as well, on top of all their other responsibilities. So we'd like to thank them for participating as well. And we really couldn't do this without you, and we thank you for your partnership and your commitment to doing this together. So thank you.
Would you 1 or 2 questions? 1 or 2 questions we can take?
Is there another mic back there? I can do up here.
I got one right here. Allison.
You got it? Okay.
Yep.
Hi, yes. I know that the data is preliminary and only partial, so my question is more in analyzing the data. It's kind of a 2-part question. So the one cohort from the age range 4 to 17, that's quite large in age, and then you hit puberty. Will you be taking that into consideration in how you interpret the data moving forward? And then also, will you separate out deletion and mutation in looking at that data set to see, is there potentially a clinical significance in one versus the other, and how that will proceed?
Yeah, great questions. Like I said, this is a really rich data that we're collecting, and so once we have the final data set, we're going to interrogate it as many possible ways as we can, and one of those certainly is mutation. We know there's differences in the clinical presentation of a deletion versus mutation. We have very few mutation participants. There were 9 in the trial, so it'll be a little harder to come to a lot of conclusions with that, but we certainly will look at deletion and mutation separate. We understand from the literature that they may have different responses, and they're coming at it with different baseline levels of functioning, so we'll look at them separately. And then in terms of puberty, yeah, that's a good question. I don't know if you noticed on there, but we had, in the 4 to 17-year-old range, we primarily had 4 to 12-year-olds. We only had 1 17-year-old. So most of the data set in that early group is from prepubescent adolescents. So we'll be able to take a look at that, at least prepubescently. We won't have a lot of information during that adolescent period, and I think that's an area we'll have to do better about getting more participants and information on that group so we can understand that better.
Hi. I would like to know if you have plans to expand test sites for next stages into new territories, into new countries, into new locations, and if so, how can we, families, help you do that?
So there won't be an expansion for the current HALOS trial, if I understood your question correctly. So that trial has completed enrollment with those 11 sites across the 6 countries, so that one won't expand. Clearly, a phase 3, no matter who runs a phase 3, they're going to be larger trials, so I would imagine there's going to be expansion in a phase 3. Different countries, greater number of families, but not in the HALOS trial.
Hi, thank you for your talk and the exciting data. So you showed that about 70% of patients showed the reduction in the delta power. So I was wondering, do you guys have any sort of early hypotheses as to what are some of the correlates of collection or correlates of disease correction? So is it correlated to genotype? Are there intrinsic factors? Could it just be, you're kind of early in the detection period and you may expect that number from 70 to go up to, 80 or 90? Or if you have any thoughts about that?
Yeah, we haven't done the correlations. Too early to do that, and we think we might need a longer time. This was a data cut from after 3 doses early on, and so it's encouraging that a large portion of them have shown something in that short period of time. So I think we will do the correlations eventually. I think the paper that came out a couple of years ago showed that that's a likely possibility that we'll see that correlation, and we'll certainly do that when we get the final data set.
I have one last question, and then we'll move on, which what I'm going to ask, because the whole community knows what I'm going to ask.
Is this a question you've already asked me?
Yeah, and I'm going to ask it again because everyone online is asking,
That's
and they're
okay.
texting me this question, so I'm going to announce it loudly.
Okay.
So the UPD ICD genotypes have been waiting a very long time and keep hearing that we need more data. And so if you can just explain to them what more data you need in order to make this decision, and are you going to be getting that data in your next evaluation of the human data you now have to make that decision, or are you saying you need more animal data, you need more cell line data? They want to know, it just says we need more data. What does that mean?
Great question. Thank you. And Allison asked me this question a lot, so thank you, Allison. So we need to analyze, for us, we need to analyze our data from the HALOS trial. We're not looking for animal data at this point. We're into humans now, and that's going to give us information that we need that'll help us make decisions about going into the really young kids, the infants, and also the other genotypes. As I mentioned, given the mechanism of action of this ASO and the genetic cause of Angelman syndrome, this ASO should theoretically be able to be effective in all individuals with Angelman syndrome. What we're looking for from the HALOS trial is if there's things like a dose response, like if you can dose response to a level of UBE3A that you think you need to see an effect. We think that's important for UPD and ID because in theory they have 2 copies of the paternal gene and you could overexpress. That's a theoretical risk that we haven't seen data to support that, but it is a theoretical risk. So we'll take a cautious approach. We'll get that data from the HALOS trial to be able to inform on that decision. And we do plan, ultimately we plan to develop a drug that treats the entire population. So that's our goal. And we have to do this in just a really cautious, systematic, and thoughtful way. And so the data that we get from HALOS will inform on that decision on how we approach it in another trial. Does that answer?
Yes. Did everyone hear that loud and clear? No more mice? Great. Thank you.